Hey guys, welcome back. Now we're officially entering the genetic unit. In this lesson, we're going to be talking about the structure of the genetic material. So let's start. By the end of the lesson, you are expected to know the genetic material, the DNA packaging, DNA structure, and its components. In a long lesson, we're going to be covering those points that are listed on the slide. Let's start with the genetic material. Before the discovery of DNA, many scientists believed that there were certain types of protein that carried the genetic material. And this was because proteins are structurally diverse and complex, making them seem like better candidates for storing and transmitting genetic information. And this hypothesis was true until DNA was discovered by Friedrich Miescher in 1869. And DNA, or deoxyribonucleic acid, is the molecule that carries genetic material, which can be passed down from one generation to the next, or from the parents to their children. And genes refer to the specific segments of DNA that carry the instructions for particular traits or functions. And a trait is a characteristic of an organism. In humans, traits can be the hair color or the sound of a person's voice. And this picture shows a ginger family. Their hair color of ginger is one of the human traits. And the transfer of genetic material through DNA allows inheritance, meaning the traits of parents can be passed down to their offspring. As we mentioned in the previous slide, organisms store their genetic material as DNA. And actually, for most of a cell's life, DNA exists as chromatin, which is a cluster of DNA and proteins, and that is invisible under light microscopes. And the DNA chromatin is tightly coiled around proteins called histones, forming nucleosomes to maintain structure and prevent damage. And actually, nucleosomes are present in both chromatin and chromosome. So what is a chromosome then? Before cell division, chromatin condenses into chromosomes, organizing DNA and ensure accurate genetic transfer. And when a chromosome replicates, it doesn't turn into two separate chromosomes. Instead, it replicates to two identical sister chromatids that are still considered one chromosome, and we call them duplicated chromosomes. And these two chromatids are connected at the centromere. DNA has a form of a spiral, or commonly known as a double helix. And DNA consists of a long chain of subunits called nucleotides. And a nucleotide has three parts. The first part is a ring-shaped sugar called deoxyribose. And the second part is a phosphate group. And the third part is a nitrogenous base, which can be a single or double ring of carbon and nitrogen atoms. And there are four types of nucleotide in DNA, which differ only in their nitrogenous bases. The bases thymine T, cytosine C are single ring structures called pyrimidines. And adenine A and guanine G are larger double ring structure called purines. Do not worry about this, we're going to cover that in the future lessons. And nucleotides are joined to one another by covalent bonds that connect the sugar of the nucleotide to the phosphate group of the next. And this repeating pattern of sugar phosphate is called the sugar phosphate backbone, which we are going to explore in the future. Over the course of a cell's life, it will go through an orderly sequence of events known as the cell cycle. The cell cycle consists of two main stages, a growing stage and a cell division stage. And the growing stage of the cell cycle is called the interphase, which consists of G1 as in G2 phase. And during the interphase, the cell carries out its metabolic processes and performs its regular cell activities. And the cell may spend as much as 90% of the cell cycle in interphase. And during this stage, the cell prepares for cell division by increasing its protein supply, creating more cytoplasmic organelles and growing in size. It is in interphase that the DNA of cells' chromosomes are duplicated. And this period of interphase is called the S phase, and S stands for DNA synthesis. The interphase periods before and after the S phase are called G1 and G2 phase, respectively. Over the course of a cell's life, it will go through an orderly sequence of events known as the cell cycle. And the cell cycle consists of two main stages, a growing stage and cell division stage. And the growing stage of cell cycle is called interphase, which consists of G1, S, and G2 phase. And during interphase, the cell carries out its metabolic processes and performs regular cellular activities. And during this stage, the cell prepares for cell division by increasing its protein supply, creating more cytoplasmic organelles, and growing in size. And it is in interphase that the DNA of the cell's chromosomes is duplicated. And this period of interphase is called the S phase, where S stands for DNA synthesis. And the interphase periods before and after the S phase are called G1 and G2 phases respectively, and G stands for the gap. And the stage of the cell cycle where the cell divides is called the M phase. And the M phase includes two processes, mitosis and cytokinesis. And during mitosis, the nucleus and the duplicate chromosomes divide and they're evenly distributed between two daughter nuclei. And the concept of mitosis will be discussed in the future lessons. And cytokinesis is a process where the cytoplasm is divided into two. And cytokinesis usually begins before mitosis is complete. In animal cells, cytokinesis occurs along a cleavage curl, which is a ring of protein. And as a result, the cell membrane pitches off the cytoplasm into two halves to form two cells, as is shown here. And in terms of the plant cells, they have cell walls surrounding their cell membrane. And as we know that cell walls are very rigid and strong, so they must use a different and alternate process to divide. And a structure called cell plate forms inside of the plant cell undergoing mitosis, as it shows here. And the cell plate contains new cell membrane and cell wall material. And as the cell plate grows, the daughter cells form on either side of it. And eventually, each cell is bounded by its own continuous membrane and its own cell wall. And once cytokinesis is complete, whether it's in animals or plants, two genetically identical daughter cells have been produced. And each daughter cell has a single nucleus surrounding cytoplasm and organelles and a plasma membrane. At the end of the lesson, I've got some questions, so please answer them using your knowledge. And yeah, that's about it. Thank you for listening. And again, this is only an introductory video about the genetic, and we'll dive deep into it in the future lessons.